The broadcast is now starting. All attendees are in listen-only mode.
Hi, good afternoon, everyone, and thanks for dialing in to the latest HDSA research webinar series. Today, I'm really honored to have Dr. Neil Aronen as our guest presenter. Dr. Aronen is from the University of Massachusetts Medical School, but before I introduce Dr. Aronen, I just want to give everyone some quick comments if you're new to the research webinar. We hope that you'll ask lots of questions because this is a very exciting topic for the HD community about gene silencing of Huntington. To do so, in the right-hand side of your GoToWebinar screen, you should see a pop-out box that says questions, where you can just type whatever question you want at any time during the presentation. And at the end of Dr. Aronen's webinar, we'll have a chance to read them and have him respond. Also, just keep a note, too, that if you have friends or family that you think would be interested in this webinar, we are recording this, and it'll be available on our HDSA webpage within a week after today. And I just want to mention that we do have research webinars scheduled for the next 2 months. In April 21st, we'll be hearing from Dr. Ray Dorsey from University of Rochester. Well, he'll be presenting a summary of the recent Reach to HD or PBT2 trial results that came from Prana Biotechnologies in Australia. And then in May 22nd, we'll be hearing from Dr. David Howland, who's a research director at the CHDI Foundation, where he'll be speaking to us about the latest progress being made to develop interesting HD animal models. So without further ado, I want to introduce our guest, Dr. Neil Aronen. Dr. Aronen did his undergraduate work at Duke University and proceeded to medical school at the University of Pennsylvania. Following internship and residency back at Duke University, he performed his fellowship at Mass General Hospital and Harvard Medical School. And after that, he was settled at the University of Massachusetts Medical School, where he's been since 1981. Dr. Aronen is a professor of medicine and cell biology. He's the chief of the endocrinology and metabolism department at UMass. He's also the co-director of the Neurotherapeutics Institute. And last but not least, he's one of our members of the HDSA Scientific Advisory Board. So Dr. Aronen's work focuses on developing gene silencing therapy for Huntington's disease using things like RNAi or RNA interference, as well as using viral delivery of allele selective microRNAs. And Dr. Aronen will be talking, I'm sure, a lot about what these things are. On a non-science note, since we are in the middle of March Madness, one thing you may not know about Dr. Aronen, but I've come to find out after working with him for a number of years, is that he is a world expert on all things Atlantic Coast Conference basketball. So if you have any other questions about some picks that you want to make for the remainder of the March Madness, feel free to ask them. I'm sure he'll have some guidance for you. So with that, I will turn it over to Neil.
Okay, well, thank you, George, and thanks everybody for joining us. By the way, I did pick Duke to at least get it to the second round. They didn't make it this year, so so don't ask me too much about the basketball this year. So today we're going to talk about RNA silencing, and it's a way to change how genes work. So it's really basic. And Huntington's disease is a genetic disease. So that means that an abnormal gene causes an abnormal messenger RNA to be made, which then causes an abnormal protein to be made. It's a relay. It would be like a track relay, where someone starts off and hands the baton to a second person, who then hands the baton to the third person. The first person would be the gene, and then handing off to a messenger RNA, and then handing off to the protein. And then the protein does all kinds of different things in the cell. The Huntington protein is very important for different kinds of cells. In fact, if you didn't have the Huntington protein, then organisms wouldn't be made because it's lethal. So we need Huntington. There's nothing wrong with it. And even the mutant Huntington gets you to a certain point without any problem. It's then in the adult, problems do accumulate, so people develop the movement disorder or cognitive or thinking problems. So the slide you see here is a slide of the people in my lab and the people who work with me. And you see a lot of people because we're a team. Anyone who does the RNA therapy, especially in the way we do it, needs a lot of expert scientists to participate in the research process. I get research support from mostly NIH, and this is called an R01, and this is also another R01. This one is a translational grant, and this is a basic grant. The CHDI, you know well, and the CHDI funds us, and we have many investigators who work in my lab and adjoining labs that get sponsored by CHDI. And last year, Lundbeck helped us with some of our sheep studies. And I'll be talking about the sheep studies in just a few moments. So the big picture here is that we want to find the cause of Huntington's disease, and we now know that it's a genetic disease, and one gene from either mother or father that's abnormal is enough to cause the disease. We want to direct the treatment to the cause. So basically, it's a genetic disease. Let's treat the abnormal gene. We want to make sure the treatment is safe. And what you're going to see is that we can treat it in mice, but that doesn't mean we can necessarily treat it in humans, because we have to also show safety. That's one of the main hurdles that a lot of therapies have to overcome. We want to ensure that the treatment works in affected cells, so we've got to deliver our treatment to the right cells. Now, this is a brain disease, so we have to get it into the brain and in a certain area called the striatum. Also the cortex. So those two areas are critical. And then we want to make the treatment lasting, if possible, a lifetime. And we want a lifetime to be decades of life and good quality life. So here's a picture of what we're going to call a brain cell. It's called a neuron. And these various descriptions tell us how the mutant Huntington is working in certain sets of neurons. They change how the DNA makes messenger RNA. That's in the nucleus here. They change how the mitochondria work, and these are parts of cells that make energy. You don't have energy, you don't have life. There's certain organelles or small particles that pick up material from the outside of the neuron and bring it inside, and that's called vesicle recycling. All neurons have axons, and these axons communicate with other neurons, and these are impaired. So information doesn't proceed from one cell and then to another cell. There's altered autophagy, and this basically gets rid of waste. So waste can accumulate, and some of the proteins come together called aggregation. And there's also a cortical problem, another area of the brain, and here a growth factor called called BDNF. Doesn't get delivered from the cortex to the striatum, and the striatal neurons, these cells, suffer by it. So you have all these different ways in which the mutant huntingtin, the protein, causes problems in the neuron, any one of which would be enough to cause cell death and also abnormal function of the neuron, any one of which, and you got all these. So we have to go a step back and try to reduce the amount of mutant huntingtin that gets made. If we took care of vesicle recycling, we still have problems with the mitochondrial dysfunction. Sort of like my old car. You fix one part, I'm in the shop for something else. So in what I'm going to tell you, there are various ways to change how the messenger RNA, which is one of the avenues of gene function, how it makes the mutant huntingtin. And we use a technique called RNA interference or RNA silencing. It actually was discovered by my colleague who's right upstairs from me. And he discovered it and reported it in 1998, and he won a Nobel Prize for it in 2006. That's only 8 years. And it was only 8 years because it's so important. Now, what he found was small RNAs, and an RNA is just a chemical within cells, can be used to kill the RNA itself, a larger RNA. So small RNAs can be used to target and inactivate large RNAs. Very powerful. And we can also put the small RNAs and package it into a virus. Now, the virus we would use is a safe virus, and we use a virus because the virus has important characteristics. One characteristic is that it gets into a cell, it gets into the nucleus, it resides in the nucleus for years, sometimes 10, 20 years. And the idea is that it makes these small RNAs that seek out the mutant huntingtin RNA, and it does so for decades. And so in theory, it can reduce the amount of mutant huntingtin that's made for decades and provide a treatment for decades. And you only need to get the virus into the neuron once. So that's the promise of the system. So we have two types that we can package, two types of small RNA. One is called an oligonucleotide. It's an RNA. And the other one is a microRNA. And we don't need to go into the details of either one of these. But we need to know that it can be packaged into the virus that we're going to work with and therefore delivered to the neuron. The virus carries it through. It's almost as if it were a Trojan horse. So inside the virus are these RNAs that are therapeutic, and the virus gets in and then can make and release these small RNAs, the contents of the Trojan horse. And this is a picture of the virus, and it has all kinds of parts. But right here, these two loops are what we're going to focus on. So these loops contain within them the small RNA. It's only a small part of the entire viral package. But these get cut, one here and one here, or one here and one here, and have RNA that's here and here on either side, and this gets released, made in the nucleus, and then released into the rest of the cell where it can block making the protein. And this virus, the small RNA is in this side, It's just the way we construct it. And in this virus, it's at another place, here. And the virus gets made because there are regulators of the virus that can be active. One is called pol2, and one is called pol3. The pol3 makes much more than the pol2, but both of them might be clinically useful. So here's an example of the how we can use these viruses. So we made a virus. We call it adeno-associated virus. It's similar to, but not like an adenovirus that causes a cold. But it doesn't. The adeno-associated virus is fine. And we put it into the circulation, and we know that it gets into the liver. 1 month after the injection, we measured the mutant Huntingtin messenger RNA. We also measured the mutant Huntingtin protein. And you can see, and these are different varieties of the small RNAs, you can see the extent of knockdown in the liver. They average about 75%. That means that 75% of the protein that would ordinarily be there, the mutant Huntingtin, isn't there anymore. It's eliminated. Now, if Huntington's disease were a liver disease, we'd be nearly finished. But it isn't a liver disease. It's a brain disease. And this is an example in the brain. Same idea. So we injected the virus that contains these small RNAs into the striatum. That's this area of the brain. This is a mouse brain. So, and then we took out the a part of this mouse brain. And what we find is that for protein and messenger RNA both, the Huntingtin gets knocked down. And it gets knocked down for the protein about 50%, and it gets knocked down for the messenger RNA about 50%. 50% knockdown is enough for a therapy. We have in other studies that I'm not going to go through, we know that we can reestablish normal cell function by eliminating 50% of the mutant Huntingtin protein. So we know that this RNA silencing can work. Here's another example. So we did the same thing, and we knocked down the mutant Huntingtin. This time, it's a true mutant Huntingtin, and this animal, this YAC128, has the mutant protein in it. Same protein, it's a human protein, same protein that causes the disease in humans. And you can see here, we were able to knock down about 50% using the virus with the small RNA, about 50% of the mutant Huntingtin. So so far I've shown that it works. It knocks down 75% in the liver and about 50% in 2 different studies in the normal Huntingtin and the mutant Huntingtin. So in order for this to work in patients, in you maybe, we'd have to make sure it spreads through the brain. Now the mouse brain's really small. It's maybe a gram, not even. The human brain is 1,400 grams, and the sheep brain is about 140. I got that from Wikipedia, so I know it's true. So we have a viral delivery team, and it's I organize it, but there are many people who do the work. Richard Mosier is our chair of the neurosurgery department at UMass. Matt Gunas runs the radiology research. Denise O'Connell is a veterinarian. Guangping Gao runs the gene therapy core. And Marion DeFia is at Mass General Hospital, and she's an expert neuropathologist. So this team is working on sheep. And the reason we use sheep is that there's a transgenic animal in Australia that has the mutant Huntington gene, the human mutant Huntington gene. In fact, let me just say, we just injected in January, I say we, Richard Mosier did, 40 sheep. 20 were harvested a couple weeks ago, and we're beginning to analyze the data, and 20 more will be harvested in July after 6 months after the injection. And we're looking for safety, and we're looking for efficacy of knockdown. Here's an example of what we do. This rod here is used, it's hollow, it's a cannula, to inject the sheep. This is the sheep head, and it's going right into the striatum. And we use this to lower this gadget to lower the cannula. We keep it in for about 40 to 60 minutes, and it's a slow infusion. And then it has time to spread in the area, and in the virus in the area that then gets taken up by the brain cells, where it goes to the nucleus and makes the small RNAs that will silence the mutant Huntington gene and messenger RNA. And these are examples of the spread. This is in micrometers, microns. And so it has pretty good spread through the striatum, pretty good. Okay. So that's good. It gets into different areas, anywhere from, let's say, 40 to 60%. And this is an example of what it looks like. Now, GFP is a marker. It's fluorescent, so you need a special microscope to look at it, and it's green, so that's why it's called green fluorescent protein. And you see it gets into neurons. These are all neurons. Nu N marks neurons. That's why it's called Nu N for neuron. And that's in red. And then we get these orange looking that shows double labeling. That means the GFP of the virus is in neurons, and they look orange. Not all neurons take it up, but many do. And this is another example. One was on one side, one is on another. And these are in sheep, so we're getting reasonable spread. Now, you might ask, how many neurons do I need to get the virus in for a real treatment? And we don't know an answer to that. But we do have an idea that if we get it into 40 or 50% of neurons, we would expect a reasonable therapeutic response. And that's what we're getting right now. And this is more information on it. So we get good spread, but they don't always go into the cells we want. This particular variety of virus, called RH10, gets mostly into glial cells, another type of cell in the brain, rather than neurons. It gets into only a minority of neurons, whereas other types in other packaging paradigms can get into 35, 45% of neurons, much better. So we then have to select the best types of treatment, and we've got to show that it's safe. So how do we do that? Safety is a really big concern here, because once we get a virus in, we can't exactly take a forceps and get it out again. And a critical question is how we might be able to modulate the messenger RNA for mutant Huntington in other ways. So for the safety, we're really relying on the SHEEP project. And David Howland at CHDI, who will be speaking with you, has a really good handle on it. He's helping coordinate that study. But we could also use a small RNA that we get into the brain by just infusing it. So there are advantages to that. When we put a virus in, we can't take it out again. It's there. If it works and it's safe, it's a first pass for a therapeutic because its effect lasts for decades. But we may not get it into all the cells, all the neurons, and we might want to target the neurons that are void of the virus. And so we can use what I'm going to call chemically optimized siRNAs. These are small interfering RNAs. This is actually what Craig Mello, who won the Nobel Prize, this is what he discovered. And we packaged some of these into the virus. But this is what he discovered. And what it does is it binds to the RNA and with some proteins, and it prevents the RNA from making the protein that causes disease. So there's a block here. These are examples of small RNAs that we tested. And in the red are some that are called hyperfunctional. They're very potent. So at a very low concentration, these will work. And they'll work in tissue culture. And here's one where primary cortical neurons, and you can see some of these small RNAs knock down Huntington 80%. So they go from, this is a dose response. This would be 100, and they're down to 75, 80% knockdown. That's quite sufficient for a treatment. So we have the small interfering RNAs that are potent, so we've made those. We can package them into some small vesicles. They carry information from cell to cell, so we're looking at that to see if that could be a delivery system for us. And then we want to show safety. So how much is Huntington is effective and safe? We estimate that 50% of the knockdown, or 50% of the mutant Huntington that is around after treatment should be enough for normal neuronal function. So the brain cells will work just fine. That is, we don't have absolute data on that, but that's where the data are pointing. Our next studies will be to figure out the best types of virus, adeno-associated virus, AAV, that we should use. And then we have to change the virus in a way where it works really well, but it doesn't overproduce the small RNAs. That could be a problem too. So we don't want overproduction. We want just the right amount. And then we're looking at other delivery systems, including those small vesicles. So one of the grants is on that. We want to work with the hyperfunctional or highly potent siRNAs. And we want to ensure that the mutant Huntington, but not the wild type, as best we can, gets silenced in a therapeutic manner. And we're back to the lab. So we have about half an hour, and I'll be more than happy to answer any questions regarding these therapies.
Thank you very much, Neil. Yeah, we've got quite a few questions coming in, so please, if you have any other questions, take this time to type them in the questions box and send them to us, and I'll read them to Dr. Aronin. While people are typing, I have a question for you, Neil, and that is, it comes up over and over again, this idea of where should we be targeting the gene silencing? We all know the striatum looks preferentially degraded in Huntington's disease, but there's also the idea that we should, maybe the striatum is a victim of a poorly behaving cortex. Are you doing any work to try to figure out if we should be infecting the cortex of sheep or mice versus the striatum, or both?
So I think ultimately we need to do both. We need to treat the cortex, because remember I showed that this growth factor is coming from the cortex to the striatum. So we don't want to eliminate the growth factor. We want to make sure it's maintained. In our initial studies, where it's focusing on the striatum as much for safety as for efficacy. So we want to see the knockdown of the mutant protein, but we want to make sure it's safe. And rather than go all over the brain with that, we're focusing on the area that's affected first and foremost. However, ultimately, we're going to need to treat the cortex and striatum and consider them as a package, because they're part of a relay too. We don't want to drop the baton between the cortex and the striatum.
Great. So here are a few questions, Neil. One you may or may not have insight into, but maybe just your best guess, if you feel comfortable doing it, is the question is, when do you think, when will this approach become viable for clinical trials in humans?
Well, let me answer that, because I don't have a specific answer, a date. Mm-hmm. What we're doing in the sheep is trying to show safety. So we're using a particular virus for delivery. If it turns out that it's safe, or if we have to change promoters or other parts of the virus to make it work better, if that's all safe, then we will probably go to the monkey for just a safety study, not for an efficacy study. And my sense, and I'm not the FDA, so I don't make policy, is that we'd need a few monkeys, maybe fewer than 10, to show safety. That means we look at the brain, we make sure that neurons aren't damaged, there are no holes in the brain, that sort of thing. The animal moves appropriately, just to make sure we're not doing any harm. And once we have that, with the information in the sheep and the information in the mouse, then I think we can start talking about a phase 1 trial, which is safety again. The safety keeps coming up. So we are beginning to analyze the sheep data. We have other sheep that we will probably be treating. And so that's going to take a couple years. The monkey studies might be done in parallel, so at the same time. And that would be good. So minimally, I would say 3 years. Here's the problem. If we rush it and there is a safety issue, then we could be harming people, and we don't want to do that. I know that there's a push to treat as soon as possible, but there are lots of studies where you rush too much, it actually delays the therapy because you have results you hadn't expected.
Mm-hmm. That's great. Another question is, and I think you may have answered this, but maybe it'd be worth addressing again. It's a question about what virus is used. Maybe there's some concerns about hearing a virus going into your body or the brain, but can you just tell us again why the AAV virus is safe?
So the adeno-associated virus, which comes in lots of different flavors and types, is already being used in humans. It's being used in gene therapy, adding genes in. It's being used for treatment in Parkinson's disease to add in growth factors, for example. Many of us have had infections with adeno-associated virus and didn't even know it. In fact, the sheep that we're treating in Australia, probably, if almost all of them, probably have had some adeno-associated virus infection before, because we can measure antibodies against it in the sheep. We have drawn blood from the sheep, and we see that they've had infection with the adeno-associated virus. It is not known to be a harmful virus. It does not integrate or insert itself into the genome. It actually resides outside of the genome, where it makes the small RNAs. So as far as we know, and now this is approaching a decade of research in monkeys and in humans, this virus is safe. So I can't go beyond the 10 years because we don't have the information. But that's pretty good.
Great. Another question, Neil, is to date, for what condition other than HD has RNA silencing achieved the most progress in combating disease or onset of disease? Are you familiar with any other progress?
Yeah, there's some eye diseases where it's been used. It's still a new therapy. There are some companies that have used the small RNAs called siRNAs, and these companies have found that it changes some liver proteins. You can see how the liver likes to take this stuff up. So it changes some liver proteins, and these are in phase 2 and phase 3 studies. Mm-hmm. This is a new therapy, so that it's hard to have a whole wealth of experience because we're making the experience.
And I think everyone is very familiar with ISIS and their antisense technology. They have a drug recently approved, you're right, Neil, for a retinal degeneration. I forget the exact disease, but it is approved for, it's a gene silencing approach for an eye disease. And I think there was a recent liver disease that was approved last year as well.
That's right. That, I think, was Alnylam, another company. So these companies are very enthusiastic about these small molecules that are cutting back on the RNA, silencing them, and we're just we're just beginning to understand the breadth of possible therapies because it's new.
Right. Maybe you kind of touched on some of these answers, but here's a question regarding what's the main challenge in your mind in getting antisense or knockdown therapy into human trials, and when will that be? You've kind of answered that. But is it safety? Is that still the main challenge for us right now?
Yeah, I think safety is the main thing. Now, we still want to use these large-brained animals like the transgenic sheep for study because we still want to know how many neurons will be treated. You know, if you're going to treat you're going to treat a region of the brain, you want to make sure you get to every aspect of that the brain. But unless we show safety, it will never be approved, and large companies won't pick it up to do more research on it. And the same question comes up because it's so important. We've got to show that it gets good spread, it does its knockdown, it doesn't do too much knockdown, doesn't affect other genes, and the neurons will not be adversely affected. Mm-hmm. But there's something in that. A lot of the people who study this sort of therapy do a month or 2 months. That may not be long enough. A person who gets treated with either the virus or with the siRNA itself and would need multiple treatments is going to have to take this for years. The virus is going to have to work for years. The siRNA or or antisense oligonucleotide or some similar type molecules will have to be given for years. 1 or 2 months isn't going to do it. We need to have longer-term treatment. That's why we're treating the sheep for 6 months. If we see something there, we may have to extend the time for treatment. This is going to be meaningful. We got to do it right.
Mm-hmm. There's another question regarding different types of viral vectors, and is there one viral vector better versus another, for instance, lenti versus adeno?
It's an excellent question. There are two. The lentivirus can also get into the brain if it's delivered and gets into neurons. There's some differences, though, between the lentivirus and this adeno-associated virus. The lentivirus gets right into the genome, so it can actually disrupt genes that make proteins that may be very important. So it may insert itself into several places in a genome, so it could disrupt multiple genes. It also doesn't spread quite as much as the adeno-associated virus, so there's a practical matter. On the other hand, it's a reasonable delivery system given those provisions. The adeno-associated virus comes in many flavors. There are 10 that are currently used in experiments. Dr. Glial, who's an expert in this, he runs our gene therapy core, has another 190 that he could be testing. So there are a lot of these. The one we are focusing on now is AAV9. We have tried the RH10 and 8 made in rhesus monkey. That's why it's RH. And they get in pretty well, but the RH10, which I thought would be great because it spreads so well, gets into glial cells. Glial cells are another type of cell which is not fundamental to Huntington's disease, but it may be affected later in the disease. So we're focusing on that, and we're also looking at another AAV type. So we've got to test those to make sure they work well in a large brain. We also realized that some of the viruses work better in sheep and might not work as well as monkey, or vice versa, work well in the monkey and not in sheep. So we will need to test the monkey. And we're really making a leap of faith that if it works well in monkey and sheep, that it'll also work well in humans. We have some information that some of these would work, but they're the older viruses and don't spread very well. So we've got to balance what we're doing with the safety, but also what may work the best.
Here's another question. It's a great question that comes up a lot. Is there a concern that RNA treatment could knock down proteins other than the mutant Huntington?
The answer is yes. We have to make the small RNAs as specific as possible. Even if they work, they affect other gene products or other proteins, that may not be a problem. But we have to see how much they would silence other gene and gene products. We take lots of medications for problems that have effects on messenger RNA. But unless it really provides a real change in the messenger RNA or a change in the protein that affects how cells work, it may be a measurable change, but nothing that's really therapeutically important. But we have to, that's part of the safety issue. Mm-hmm. So that's why we're being careful. Part of what we're measuring in the sheep is something called gene sequencing, or mRNA sequencing. And that is, we're going to measure a whole host of RNAs to see what changes. And we're also matching it with behavior. to make sure that there's no abnormal behavior that's caused. Great question.
Here's another one related to the sheep, Neil, what is the effect of the virus on the sheep immune system? Is that something you guys are interrogating?
Well, we're looking at antibodies that are made against the virus, but we already know that there's some, even before we treat, that they have antibodies against it. We also are looking at the sheep, not the ones in Australia, but actually sheep that we purchase and that are in our animal medicine department, and we inject the AAV into those, and we look for pathological changes that are representative of the immune system. So that means certain chemicals that increase after inflammation. We look at the neuropathology to make sure that the neurons are healthy and that there aren't immune cells that collect in the brain that's treated. So that's actually part of the safety issue.
You touched on this. You mentioned glial cells briefly. There is a question about glial cells. And do you predict the glial cells may be involved in pathology? And do you see any benefit from targeting glial cells that uptake mutant Huntingtin in addition to neurons, or uptake, I guess, the virus in addition to the neurons?
So there is almost, or at least as many glial cells, these other cells, as there are neurons. They're everywhere in the brain. And the glial cells have a lot of different responsibilities in the brain. One of them is to exchange chemicals with neurons. They're almost like a nourishment source for the neurons. Another type of glial cell called microglia accumulates in the human brain with Huntington's disease around neurons. And we think they're accumulating to try to clear the brain of the neuron because the neuron's damaged. There's also information that these microglia can help the synapse. That's a communication area of neurons to help stabilize the synapse, something that's opposite. So these microglia have a lot of different roles in the brain. And we've done studies. We've done it with Marion DeFilia at Mass General on quantifying the microglia in the HD brain. And we know that they're increased. I don't know that it's a good idea to squash the activity of microglia, and the reason is that some of the microglia might be cleaning up neurons that are damaged. That may be good, and some of them may actually fortify the neurons so that the synapses work better. We don't know enough. Now, there is a school of thought that the microglia might have a critical role in the pathogenesis of Huntington. So once there's an abnormality in the neuron, the microglia have a role in advancing the Huntington's disease itself to make it worse. But it's a theory. When you see the microglia just in a slide, you don't know how they got there, and you don't know what they're doing. You just know they're there. So right now we're using a virus that treats actually the neuron and some glia. It gets into both. So if there is some mutant Huntingtin that's causing the disease from the glia, those can get treated too.
Great, thanks, Neil. Here's another great question in regard to the number of times a viral treatment might be necessary. So within mice, one treatment might be, or appears to be enough. But once in the larger human brain, will more treatments of a viral delivery be necessary? In other words, is there a potential is this a potential silencing that will require long-term viral injections into the brain?
Another really good question. Actually, we would be reluctant to treat more than once. And the reason for that is that you might get an immune response to the virus with subsequent injections of the virus. So you want to treat once, get really good spread, uptake into the neurons, maybe the glia, and that would be the viral therapy. We don't think that using the same or different virus, let's say an AV, the AAV9 and an AVRH8, is a good idea because they do share some of the same proteins that can elicit an immune response. So my feeling on that is we got one chance. Let's do as good a job as possible. Those in the field are really reluctant to keep treating it over and over because they think that's a good way to get the immune response activated.
Great. More questions,
George,
we have lots more. We have 5 more minutes. I'll try to get through as many as I can, and I apologize if we don't have time for all of these questions.
I can stay a little later if you want.
Great. Here's another one. Drilling a hole in a patient's head seems a bit invasive. Are there other approaches for delivering the virus to the brain?
Well, we're working on them. We're trying to get the virus to cross the blood-brain barrier. We can get it across, but only a few neurons are affected, you know. So we're not going to be able to treat enough neurons right now. It doesn't matter to me that the liver takes it up too. Maybe getting rid of mutant Huntington is a good idea in the liver at some point. So we're working, our gene therapy group that I listed is working really hard to try to get another viral type that crosses the blood-brain barrier. So we can treat that way also. But since this is a single series of injections, with probably several into the brain at one sitting, that I think it's invasive, but I don't think that it's such a problem that we wouldn't treat, because we're treating once for hopefully decades. Let's put it in, if you didn't have a functioning liver and you needed a liver transplant, that's much more invasive. If you needed a diabetic who needs a kidney transplant, that's much more invasive. So, and we would do that if it were necessary.
Great, thanks, Neil. Yeah, and kind of going with that, you mentioned that there's a lot of development going on behind the scenes of developing new types of viruses that maybe cross the blood-brain barrier. There's also development going on to develop viruses that are regulatable. As you mentioned, once this virus gets into the brain, it's on, and it's silencing Huntington potentially forever. And that could be a concern. So your group is, correct me if I'm wrong, but is also working on ways to shut off the silencing once it's in the body.
That's right.
Which is
That's right. So the way we want to do it is to change the gene so that it's turned on if we gave a drug, and it's not turned on if we don't give the drug. And I think that's, you know, the people working on it are hopeful that they'll be able to do it, but there are a lot of technical obstacles to overcome. So we're working on it. The way it's being used experimentally is there are 2 different genes that have to be put into the same neuron for this to work, and we're trying to get it so that it's in one gene as part of the same gene that has the small RNA, So it's one package that goes in, and the medication we would give would turn it on. The medication itself may have nothing to do with Huntington's disease. Its sole job would be to turn the gene on or off because it's in the virus. So maybe in a few years we will have that available to us. But we have a group working on that.
Great. Another great question is coming in where they want to know, would this gene silencing approach be more effective for someone who shows symptoms or someone who is pre-manifest HD or both?
Well, I have an opinion on that. The opinion is that the sooner we can treat, the better. This is not a way to cause regrowth of brain cells. It doesn't do that. It hopefully will salvage and protect the cells that are already there. So we would, in my view, the earlier the better. Now, it could be in the first phases of the trials, we'd have to wait until we have a presentation of Huntington. So a chorea, movement, or some depression or something like that. But ultimately, I would rather treat earlier than later.
Great. Since maybe there's another one where, another question here where it's more of maybe more of an opinion question. From your expert opinion, would you see this knockdown therapy as a more viable approach than stem cell therapy?
Well, they're two different approaches. So I'm a clinician too, and I'm an endocrinologist. That's a long story of how I got into Huntington's disease. But many of our therapies require multiple drugs and multiple approaches. So I'm used to giving multiple approaches. I think Huntington's disease will have multiple approaches too. I view the silencing as a basic approach with the virus, and maybe supplemental with the small RNA as an infusion. And stem cells might also be, but it's not only putting it into the brain, but the stem cells have to develop into mature neurons and integrate information in the brain. And that's no easy task. The same with the iPS cells, these other stem cell-like cells. So I think there are multiple meds that can be used at some point, and we're going to have to be able to handle all of them, as we do with diseases like diabetes and high blood pressure, where it's usually multiple approaches that work.
Mm-hmm. Great. Well, we'll have time for one more question. It's a really knowledgeable question about what's a study that maybe you're aware of, Neil, that was published late last year. And this question regards to what can we take away the community from the ALS study in humans that was done with antisense therapy. So I think this person is referencing the recently published paper where ASOs, or antisense oligonucleotides, against the SOD1 gene. This is a gene that causes ALS or Lou Gehrig's in a small subset of ALS patients, was given to people and found to be safe.
Well, I think it's a promising therapy. I think the location of the treatment is important. In ALS, they treated the spinal cord, which is right there, and the ASOs can get right in. When the ASOs were given to monkeys, and this was published a year and a half ago, they got into spinal cord great, but it didn't even get to the striatum. There was very little uptake, and that was in the monkey, which is, again, a much smaller brain. So I think it shows that these small molecules that knock down the RNA can work. And the monkey study shows that. The human study is a whole nother thing, because you'd have to show that there's improvement in the patient. And the monkey, they showed basically that there was knockdown of the SOD1. And so that's still a study that is ongoing to show real improvement. But I think it's very encouraging.
Yeah. And it's encouraging, the people know that this study, this was work from ISIS's antisense technology in patients. It just showed that this approach, injecting ASOs into the spinal cord or intrathecally, is safe, and that's great. It doesn't mean it's being effective or it's efficacious in the patient yet. Those studies are ongoing. But what's promising is that is the approach, the intrathecal or spinal cord administration. That's the approach that ISIS and Roche will be taking forward for their first clinical trial in HD patients. So it's promising and encouraging to know that it's been in patients. A similar approach has been done in ALS, and it's been shown to be safe. So it's encouraging.
Yeah, but it has to get to the striatum to be effective.
So that's, we're run a little bit over, but that's a great thing because there's been lots of interest, and I apologize if there have been some questions we haven't been able to get to. If you have a burning question that you feel we did not answer adequately, please email us. You can just write us at researchupdates@htsa.org, and we can either send that question to Dr. Aronin, or hopefully I can answer it for you. So with that, I just want to thank everyone for dialing in, and very much thank Dr. Aronin for taking his valuable time to present this very exciting topic to all of us. Thank you, Neil, and thank you, everyone.
Welcome.
Take care.